This is TWIS. This Week in Science, episode number 1069, recorded on Wednesday, July 29th, 2026. The Dish on Disease in a Dish. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with pancreas, disease, and memory. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. There once was a cell from a patient. It was always useful adjacent. With more than a wish, it's now in a dish, and new therapies are possibly nascent. Learn all about it here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science, everyone. Welcome to another episode of This Week in Science, back to bring you all the science that we love to talk about, things that have happened this week, last week, within a week, and more. We want to learn, we want to grow together and understand the world and be curious together. Thank you for joining me for this episode tonight. Blair is out, and hopefully we'll be back again next week, but she's been hosting the show for the last 2 weeks without me, as I had a little holiday, which was wonderful, and it's so good to be back. Thank you all for being here tonight on all of our chat rooms. Tonight, we have an interview with Dr. Dannielle Engle, a Salk Institute researcher who studies the pancreas and organoids, disease in a dish. I've also got a couple of new stories about memory, the brain, the stomach, how it's all involved, if I can remember them. But before we jump into everything, I want to remind you that subscribing to TWIS on whatever podcast platform you enjoy podcasts are is the best way to find TWIS on a weekly basis, our wonderful edited podcast. Or you can join us live Wednesday evenings, 8 p.m. Pacific time-ish on YouTube, Facebook, or Twitch. And if you need information about our shows, show notes, links to stories, all sorts of cool things, head over to twis.org. That's also where you find our Zazzle store link and our link to our Patreon, which Patreon is really how we support this show. We are listener supported, so there are no commercial biases. So thank you for supporting TWIS. Thank you for being here tonight. Everyone in the chat room, let's have a great show. It's time for me to introduce our guest for the evening, Dr. Danny Engle. Dr. Engle is an assistant professor at the Salk Institute, specializing in the early detection and treatment of pancreatic cancer. She received her BA from Northwestern University in Biology and Asian Studies and her PhD in Biology from the University of California, San Diego. She recently published a paper in Cell Stem Cell describing a new platform using patient-derived pancreatic organoids for disease study and development of personalized treatments. And I think this sounds like a really exciting direction. And we've talked about organoids a lot on the show over the years, but I think it's really important to get somebody who's really involved in the research and use of this kind of a platform to come and talk with us about how it works, the challenges, all these things. Thank you for joining me, Dr. Engle.
Lovely to be here. Thanks for having me.
You're welcome. So first off, before we get into the study and the nut of the nitty-gritty of everything that you've been working on, how did you actually come to start studying pancreatic disorders in the first place?
Oh, so I would say that my journey has actually not been super typical for a scientist. I actually went to college for music before switching to biology. And a lot of that had to do with like, using creative part of my brain, but also figuring out that while I might not be the best test taker, I'm really good in the lab. And so for for how I got into the pancreas, I started off working in genetics, but then I moved into cancer pretty quickly because my dad was diagnosed with pancreatic cancer. And about 10 years later, my uncle was diagnosed with pancreatic cancer. And so for me, it's been kind of a personal motivation combined with this really scientific drive to understand why is it so hard to treat and find early.
Why is it so hard?
I know, right? I mean, I wish I had all the answers to that. I think that if you zoom back out and you look at the incidence of pancreatic cancer, it's relatively uncommon. So there'll be about like 60 to 70,000 new cases this year. And so it's a low incidence cancer, but it has a very high mortality. And so I think part of the problem is that means that screening a general population is not what insurance companies would call cost effective. Which is a hard, statistic and calculus for me to learn since I'm a patient advocate, but at the same time, false positives do harm. And I think that's the big challenge. And so that's also how I got started in pancreatitis, because that's a major false positive for any test that's trying to detect pancreatic cancer. So they're two very similar diseases in terms of their biomarkers, but their outcomes are very, very different.
So when somebody is coming in with stomach pains and has the etiology, the presentation of certain aspects, it could be pancreatitis or pancreatic cancer, and there's no way of knowing the difference until you go into the pancreas itself.
That's right. And so in order to confirm a diagnosis, it's not like, a simple blood test or CT scan. It does require a biopsy to really understand what the lesion is. And I think there's multiple challenges here. Pancreatic cancer itself, for mathematical modeling and autopsy studies, takes 10 to 12 years to form. So it's not that it moves too fast for us to find it. It's just by the time that there are symptoms, those symptoms are usually caused because the tumor's gotten big enough to impinge on other organs, or you're starting to get pain that's from a tumor pressing on a nerve. And so I think at that point, there's already trillions of cancer cells in your body. And at that point, 85% of the patients already have metastatic spread.
Oh, wow. So once it's there, it's at a really late point.
Yes, that's when we're finding patients.
Generally. Yeah. Okay, so if you, I say we, but if you and other researchers can find biomarkers that would be useful to discriminate between pancreatitis and pancreatic cancer, then that could allow earlier detection.
Exactly. That was the original goal. Yes.
Okay. But pancreatitis itself, over time, there can be acute. So you have different kinds, and chronic pancreatitis can actually lead to, in some cases, pancreatic cancer. Can you tell us the story of the pancreas?
Yes, we will start all the way back. And, I originally was training as a developmental biologist. And so I come at this with a unique perspective where I'm like, we got to understand what the pancreas normally does. And when I say pancreas, a lot of people have no idea where the pancreas is. Right.
It's one of those organs that people are like, you mean?
I know I have one, but it's.
Is that a tunnel station on the London Tube?
Exactly. Isn't that a saint?
Yeah.
I think that when you say pancreas, a lot of people immediately think insulin, diabetes, which is an important function of the pancreas. That's what we call the endocrine function of the pancreas, but it's actually primary function. What it spends probably like 90% of its cellular mass doing is producing digestive enzymes, and that's the exocrine function. And so when we talk about these cancers, they're the most deadly type is this pancreatic ductal adenocarcinoma. It's arising from the exocrine compartment, not from the hormone producing, insulin producing endocrine cells. Yeah. And so what the exocrine compartment is doing is it's making all these digestive enzymes, which are designed, they're proteases, so they chop up carbohydrates and lipids and proteins. And so I think that it is something where if you have digestion going out of control, it could be catastrophic inside the human body. And so the
So
exocrine
when you say
functions.
what digestion going out of you mean the acids of the stomach just continuing to
Exactly.
flow into your intestines and
That's definitely part of it. So like, the acids kind of like can like help solubilize and start breaking down, but it's really enzymatic digestion that releases a lot of the nutrients that we're absorbing. And so this enzyme part of the pancreas is a vital function. You can try and replace those enzymes after removing a pancreas, but it does decrease your lifespan to remove your pancreas by about 10 years. And
that's
so it's
significant.
it's a pretty big loss. And so that's why there's not commonly prophylactic pancreatectomies where you voluntarily get your pancreas out, because it is vital. And
Yeah.
so all those digestive enzymes, in order to keep them inactive, you have this ductal tree, which is basically a giant tube that goes through your pancreas, collecting all of the digestive secretions. And the ductal, oh, fantastic. uh, exactly what I'm talking about. And so, and the ductal cells are producing bicarbonate. And so this isn't like a, typical like sexy scientific thing to talk about bicarbonate production, but as
Oh
human
my gosh,
beings.
I was a
Okay.
physiologist. I'm
Okay. So you're
I'm
into
I'm
this. I
way
love
into
it.
acid base.
Okay.
Oh my
I
goodness.
love it. I did not
Bring
do
it.
well in chemistry, so this has been a tough journey. Like I ended up in this field because that's where the science, pointed us.
Where it
But so
led.
this bicarbonate production is actually remarkable in the human body. So think about a 2-liter bottle of pop or soda, depending on where you come from, and
Yeah.
your pancreas makes 2 liters of 140 millimolar bicarbonate every day to keep all of those enzymes neutralized and to flush them into the duodenum and then neutralize the acidic chyme from your stomach. And so that's one of the
Huge.
essential functions of the pancreas. And normally your pancreas is an absolute machine churning that out. But I think this is where we found that that function gets disrupted in inflammation and cancer.
Okay, so when the bicarbonate is not being produced, you have the acidity, you also have inflammation that comes along with it. The inflammation is going to lead to certain changes in other markers, genes, proteins, and usually we talk about inflammation as just generally being bad for our metabolism, our bodies.
Yes.
And it's going to lead to decline.
Yes, it's not exactly. That's why we've had this whole wave of like, anti-inflammatories, like, antioxidants and things like that. But it's kind of a double-edged sword. A lot of our cancer therapies actually rely on oxidative damage. And so, it's everything in biology is a balance.
Homeostasis. It's one of the best things about the way our body works. And the pancreas is essential to that. So the bicarbonate production, it's balancing out that acidity, helping to maintain the body's balance, and also helping to maintain, hopefully, lower inflammatory levels. But
Exactly.
in the cases of disease, you end up with all of these issues, right?
Exactly.
Yeah. Okay. And so, like, my dad recently was in the hospital for pancreatitis.
I'm sorry to hear that.
Yeah, it was
Okay.
very painful, and it took him out for a while, and it was kind of scary, and had no idea. Just, he went in, he said, my stomach's hurting, didn't know why. He's always had an acid stomach, right? A little bit of indigestion, whatever. And so the question is, how do we know, and is there a difference between the genetics of it, the environmental aspects of leading to pancreatitis, or even pancreatic cancer? Like, do we know how these disease states get started?
Yes. So I think it's, there's definitely a genetic component. So hereditary pancreatitis is something that was a major focus of this study. Hereditary pancreatic cancer often can arise from hereditary pancreatitis. They have some shared risk factors in terms of the gene mutations that are associated with them. And a lot of those genetic factors can actually modify your response to environmental exposures, right? And so if you inherit a mutation that makes your proteases slightly more active, and you combine that with tobacco or diesel fuel exhaust exposure, that could make you more susceptible to pancreatic damage, right? And so they all intersect. And I think that's why it's a little bit difficult to sometimes make attributions about biology and mechanism from what we call etiology. So what is the classification of the disease? What was the trigger? And so for pancreatitis, it's separated into certainly genetics, but really like toxic metabolics. So that's going to be tobacco, it's going to be alcohol, it's going to be certain medications, obstructions, and so from calcifications or ductal abnormalities, as well as idiopathic is a substantial portion. And so that just means we have no idea what caused it. And they, for some of them, it's, for acute pancreatitis, most commonly it's gallstones, but for chronic, that's not necessarily true.
And so I'm going to shift our conversation now into more towards the study that you've actually published in Cell Stem Cell. And in getting to that, you've worked for years in organoids and developing, studying organs in a dish, basically. Can you tell me a bit about the challenges to being able to create these study organs where you've got an organoid as defined as the multi-cell type, synthetic, well, kind of, it's biologically synthetic model of an organ. And we say in a dish, is it like in a tiny dish or in a big dish?
Oh, great questions. And so when we make these organoids, the mission was to make patient-derived models that are accessible to the field so that many labs can work on this same problem. And so the first challenge is, having access to the human specimens, and there are multiple different procedures that can be done that would give you access. And so being able to do human subjects research to make the models themselves is a requisite, and my lab does. But I can share the models with labs that don't do human subjects research because they're fully de-identified and it's not possible for them to be re-identified. And so at that point, I can share the model with somebody that doesn't have access to clinicians and hospitals. But I would say in making the models, the biggest challenge initially was figuring out the media conditions. And that's, again, kind of where you go back to developmental biology and you ask, well, what was required for this organ to develop in the first place? And can we recreate that niche? And so I think, that took a long time. But once we knew the culture conditions, then it's like, how do you release the cells in these biopsies? And a lot of people, when they assume that we're getting these, I always dream about working in hematological malignancies where they come as a semi-single cell suspension, but we have to enzymatically dissociate the material. And a lot of the times it comes out heavily cauterized. And so it's something where you have to release viable
scar
material.
tissue,
so much scar tissue. And that, cautery kills the cells. And so we can't make something from nothing. We need live cells. Yes. Yeah. And so there have been multiple instances where the model has failed to generate, and we can track that back to simply there were no viable cells in the specimen that we received versus other failures where we actually don't have an explanation. And so I think the important limitation of this is that we were only successful 73% of the time. So we don't know what was unique or different about those 27% of specimens that we couldn't make models from.
Yeah. And so when you're talking about making the models from cells, and this is actually written up in part of the study of how these are patient-derived models. And so this is when people are going in to have pancreatectomies.
Yeah.
Yes, when the pancreas is being removed or when part of it is being sampled or a cancer, the cancer is being taken out. You are taking advantage of that situation where people are donating the tissue.
Exactly. Yes.
Yeah.
Yes.
Okay.
And so it does require the trust of the patients, to consent for their tissue to be used for human subjects research.
And then also for the doctors who are doing the removal to know how to remove it in a way that does not do all that cauterizing damage.
Exactly. So it does require all these like very close partnerships
Yeah.
because, you know, we've done all sorts of procedures where it's like all of the organs were getting rinsed in the same PBS bucket, which is not a friend for sterility, right? And so there's all these different challenges that really goes to like relationship building with the clinicians.
I think that's another interesting aspect of the research that I imagine a lot of people wouldn't really, think about at first. How do you don't just go and harvest these pancreas to samples,
And
right? It's trust and it's relationships and it's people working together to make this happen.
exactly. And I think that like, for us, we've been as an organoid lab, we have the expertise to do it, but like, yeah, we would have nothing without the patients and the doctors. And so they're of utmost importance in this paper.
Yeah. And so you're able to get the 73% that are successful in creating the cell culture and jumping over all of the hoops through the hoops, etc., to be able to create the organoid tissue that you're creating. And then once you got to that point, what did you start looking for, and what were you trying to achieve?
Yeah, so when you're creating a disease model, the first thing that you need to do is what we call benchmarking, making sure that they actually recapitulate the disease features from the patient specimens themselves. If it doesn't capture the same genetics, if it doesn't have the same transcriptomic profiles, then you're not actually modeling the disease that you're sampling. And so a lot of work initially goes into doing what we call genetic concordance. And so it's either through some sort of whole genome or whole exome sequencing of the primary specimen that we received before we've done anything else to it, and then the generated organoid model to see how well did the genetics match. And that's where our study showed 96% genetic concordance, which in the organoid field for cancer is extraordinarily high. But here we're talking about an inflammatory disease, right? And so there was some question about selective pressures and culture and things like that. And so we were quite pleased to start off with that high benchmark metric so that we can move on to more mechanistic and biological questions.
And so once you got into the benchmark of being able to say what we're showing, this is matching. It's very, very close to complete match. And so with that kind of match, are you trying to say that hopefully you can create the kind of system where clinicians or other people could take a sample, biopsy of a patient's pancreas and put it into a cell culture system like this, and then be able to actually figure out what the biomarkers are that would allow for personalized treatment? Is that the direction that you're headed with this?
So there's a lot of precision medicine opportunities. I will say that like generating these models takes a long time. And we're not really, when you're being treated for pancreatitis, you don't want to wait 6 to 8 weeks for us to generate a model, right? Yeah. And so I think that we have to take a step back and be like, we can make generalizable conclusions based off of different genetic features or predictive biomarkers, using these organoids as a workhorse to run them through different functional tests. But really it's more of a opportunity to see which subtypes of patients respond to which drugs, and then to go back to work with the clinicians to find the predictive biomarkers to most successfully roll out in the clinic.
And so this is something that I'm going to put up the infographic from your stem cell study right now, which it shows a very brief overview of the fact that you've taken these patient samples for their various reasons for the chronic pancreatitis and created the biobank. And then there's another panel within this whole infographic that shows that there are different etiologies, but they're independent of the genotype. So basically it's genotype doesn't match phenotype, or because of whatever is showing. And what it's kind of like when I guess people studying evolutionary biology are like, ooh, morphology. And now we're suddenly going into genomics of T-Rex and other things to actually determine, evolutionary relationships, as opposed to just, oh, there was a wrinkle on the third left molar or bicuspid, as opposed to actually have genetic reasons. So is that kind of what this study has shown, is that there are genetic differences that kind of overlap?
Yeah, I think, if we go back to this idea that pancreatitis is classified by etiology, whether or not you have alcohol-induced pancreatitis or gallstone obstructive pancreatitis, that's how like patients were separated in terms of their classifications, and that would be used to inform their treatment. But what we're finding actually is that independent of etiology, there are different biological signaling pathways that show distinct molecular subtypes. So we use an algorithm called non-negative matrix factorization to let the ones and zeros tell us how many subtypes there are without any sort of bias that we introduce ourselves. Just take our data and see how many groups there are. And so that's when we first identified these 3 distinct molecular classes, where some of them are driven more by inflammatory signaling and others more by EMT and epithelial to mesenchymal transition, while some of them have very different metabolism. And so this was kind of to our knowledge, one of the first reports is really taking a deep dive into the underlying cell signaling changes and whether or not that would actually reveal different molecular subtypes that would inform how we expect those patients to respond to treatment.
And did you see overlap, or was this, someone has hereditary pancreatitis, their dad had it, their mom had it, whatever, they've got it. Is that a particular profile, or are there overlaps between them?
There are overlaps. So we find that like in these 3 different molecular subtypes, you have hereditary pancreatitis patients in each with the same mutation. So it's not like all of the patients with the mutation in the trypsinogen gene called PRSS1 are all in the same molecular subtype. And so that's what's surprising, is that the trigger that initiated the pancreatitis, and at least in our analysis and our data of these models, is not what's defining their molecular classifications.
And then one of the things that you found in common, though, even though you have these subtypes, your study said you found that some 54% were showing a mutation or some aspect of problems with the cystic fibrosis, translating, what is the whole CFTR mutant?
Yes, we just call it CFTR.
CFTR. I was trying to rattle it off my tongue, but it didn't happen. Yeah. The cystic fibrosis transmembrane conductance regulator. There we go. Which is important for that bicarbonate
to get out
passage, the release of the bicarbonate that we were talking about from the ducts. Yes.
Yes. So I think that was one of the surprising things when we started this. We were expecting to see some of the most commonly reported pancreatitis genes for hereditary pancreatitis be this trypsinogen gene called PRSS1 and a trypsinogen inhibitor called SPINK1. But what actually we found, almost, I would say, roughly 30% of our cohort had some sort of genetic alteration that would reduce CFTR levels or function. And then we also separately looked at it transcriptomically to find that there are some patients who have unexplained exon skipping in CFTR, even without the common splice site mutations. And so that's where we started to become CFTR biologists. And I'll just be completely open. I did not know very much about CFTR when we started this, but again, we kind of followed the science and learned that, you've heard of CFTR before, you've heard of cystic fibrosis.
Cystic fibrosis, yes.
Right?
But in a completely different function. Yes.
Right. And so these patients with cystic fibrosis, they have a lot of lung and GI symptoms, but in those organs, CFTR functions mostly as a chloride transporter. And so that's why you do sweat chloride tests to diagnose it, to see how functional or dysfunctional your CFTR is. But in the pancreas, it has very different types of mutations in CFTR that target specifically the bicarbonate transport function. And so the mutations are in our cohort, none of them are commonly associated with cystic fibrosis. They're only associated with pancreas phenotypes. And so this is how we had to learn, like, well, how does one measure CFTR function? And it's not like in cancer where you're trying to kill cells. So you just measure, like, how dead or alive your cells are. It's how are they functioning, right?
Yeah.
And so this was actually the kind
How
of
well are they doing their job?
exactly their normal function. And I think that's going to have been one of the refreshing things, because now we can actually go into trying to find ways of preserving pancreatic function with a lot of our anti-cancer therapies. But in pancreatitis, what we figured out is that the pancreatic organoids, basically they're like water balloons, single cell epithelial layer, and CFTR is on the luminal side. And so if you stimulate CFTR function and you get bicarbonate secretion, fluid follows. And so literally we just stimulate the CFTR function, they blow up like water balloons. They just like start inflating. And if CFTR is functional, then they will inflate. But if CFTR is not functional, if we really stimulate the CFTR function, they won't expand, they won't inflate. And that just means that there's no ion and coupled fluid transport in response to CFTR stimulation, meaning there's something wrong with it. And so that was the functional assay, which is like, encompassed basically in this very small space in this figure, but took us years to kind of like optimize and figure out how to get it to work for these organoids and to miniaturize it in a way that actually made this possible, because we're farming these organoids in 10 centimeter dishes to generate enough to do these assays. And then we do the actual functional test in a 96 well plate.
Wow. I mean, just the amount of work that's going into all the details of everything that you're discovering here, but it's just, it seems as though you went after everything, like you said, following the science, but with a fine tooth comb, and just really trying to figure out what is the thing that's breaking, what's the thing that's working or not working.
Yes.
And then
we tried to fix it.
Yes. Okay. What happened when you tried to fix it?
Oh, okay. So, I mean, there are several lines. Okay. There are a lot of FDA approved drugs to treat cystic fibrosis. And these basically try and restore function of CFTR. These mutations that people inherit, they tend to disrupt protein folding so that the protein doesn't, end up getting, to its final localization. So it's not reaching the plasma membrane. And so if it's not where it's supposed to be, it can't do its function. Right. And so a lot of these drugs are designed to like help with the folding of CFTR, help it like move to the membrane, increase the ion gating ability of these transporters. And so we took those cystic fibrosis drugs and we tried to match it, depending on the class of mutations, to the patient-derived organoids. And we were able to find in two very clear examples that if we use the appropriate CFTR agonists, we're able to then restore the swelling phenotype of the organoids in this functional test. But I think what was really surprising is, we have the CFTR mutant organoids. We expected them not to be functional, and they weren't. We had some CFTR wild type organoids that also didn't swell. And so in those cases, there's a lot of things that can impact CFTR function, like alcohol, tobacco, and a lot of this can be epigenetically encoded. So it was preserved in our models. And we were able to fix some of those too. And so there are some CFTR drugs that work regardless of if the gene is mutant or wild type. And so it kind of like opened it up to not just CFTR mutant disease, but really just in a broader picture, would improve overall pancreatic function in patients with pancreatitis.
Right. But I mean, this is kind of the interesting discovery here, because you go from understanding that there's a hereditary basis for some people, and the hereditary basis might be genetic dysfunction leading to the CFTR transporter, working incorrectly. And so you would assume that anything that is going to repair the CFTR, like the genetics from the transcription aspect forward, that would be helped. But you wouldn't expect, like you said, the repair of wild types.
Exactly. And so
And
that was
so,
that was
yeah,
unexpected. Exactly.
yeah.
But like, we took a step back as soon as we found CFTR wild type organoids not responding to this functional assay. We started looking at things like this unexplained exon skipping, or fully leveraging the suite of different FDA approved drugs or drugs that are currently in clinical trials to try and go after it. And so there are some drugs that actually improve splicing of genes, like atalurin. And so we've done sort of like some different very targeted drugs where we increase basal transcription of CFTR. And so we've come at it with kind of like a big pharmacologic toolbox that really focused on the paper and the mutation matched findings.
Okay. And so now you have models that show that there are particular presentations and particular pathways, right? So you've got these subtypes that
Yes.
don't necessarily match the way that everything's been treated so far therapeutically. Like our clinical definitions might need to change.
Might need to be updated, yes.
Yeah. And you're also showing that perhaps there is this other direction that can be gone for therapeutics for some people, depending on where they fall within these subtypes. But what is the path
What is that?
between this paper, your work, and actually having it be valuable medically to patients?
Yes. And I think this is, a lot of the struggle with people trying to do translational research is that as a PhD, I don't understand how to navigate a clinical trial, for example.
Mm-hmm.
But my clinical collaborators do. And
Mm-hmm.
so figuring out kind of like, what that path is has been kind of like, very much in our minds as we develop this project. And if you like take a look back at like how different CFTR drugs were approved for the treatment of cystic fibrosis, cell line data was sufficient for approval of new CFTR drugs by the FDA. And so there is a path based off of just of our work to think about adding pancreatitis as a new indication for some of these drugs, but also requires a lot of buy-in from the pharmaceutical companies and convincing them to prioritize pancreatitis, because it's not as many patients as the patients with cystic fibrosis. And so it's again relationship building. It's building those bridges with the people that know how to do the parts of it that you don't know, but then also trying to work with the pharma companies to get a clinical trial started.
I feel, having like come across your work, that this is a big step in the field of understanding disease in the pancreas and potentially how to approach it. How do you feel about it? Like, where do you, like, are you like, wow, this is like, are you exciting? Are you just also like, oh, what do I do next? Like, where?
Yeah, I think my lab is a little bit terrified because I'm like that meme where it's like all the push pins are connected and it's like all of these like post-it notes. And I'm like, okay, this is just the beginning.
Do you have red string going all over the place?
I should get red string and really freak them out. But I think that they've been through a lot. I won't do that to them. But I think, this is the first step, is always benchmarking the model. And we're quite happy with the data that shows that it's replicating these disease features in the ductal compartment of the pancreas. And so now how do we leverage it further? And so seeing how these processes are disrupted when pancreatitis transitions to pancreatic cancer, seeing, can we reverse that? Can we prevent that? Can we use these pancreatitis organoids to find biomarkers that are specific to pancreatic cancer, or at least try to molecularly classify them into more effective treatment groups? But we also come at this from, the fact that me and several people in my lab, we watch people be treated, or I've also had pancreatitis myself. So just knowing what it feels like and being like, this is not okay. The treatment options right now are they're not sufficient. And just understanding kind of like that this is just the beginning, because we have to let the biology teach us and go after the mechanisms that are really leading to this wound that never heals in your pancreas. I would say that when I started chronic pancreatitis was taught to me as an irreversible condition. And I no longer think that's true, but I think it's going to take us like 2 or 3 more studies to kind of convince the field of that.
And, there's probably everything is more nuanced than, we want to say everybody's going to get rid of their chronic pancreatitis. And maybe that might not be true,
Right.
right? It's different. And like you said, there are different populations, different subtypes. There's a whole lot of detail in there. But being able to understand those pathways and
Yes.
how does it move from trigger to disease
Yeah.
is
And lowering the
essential.
energy of activation of studying this problem, and so like
Mhm.
I'm the first one to admit that, like, somebody else, I'd be happy if somebody else took these models and figured out a new treatment for pancreatitis or
Yeah.
pancreatic cancer, right? And so we do share them. We are working on immortalizing them because they do. A normal adult stem cell suffers from what we call the Hayflick limit, so they will
Yes.
run out of telomeres and senesce.
But
So they're
the
not
Hayflick
immortal.
limit is somewhere around like 50 or 60, but like it actually starts going wrong somewhere around 20 or so. But
Exactly.
your paper suggested like really, really solid populations of your cells, your organoids, right, 13 or 14 generations. Is that
Yeah, so we can
yeah?
passage them roughly 15 to 20 times before
Okay.
they will senesce. Exactly. So it's
Mm.
not like, when we study cancer, because they have an activation of tumor suppressors, they become immortal.
Cancer
Turns out
is a
that's not,
different beast.
cancer's a very different beast. And
I mean,
so
that's the
in
whole
the first,
thing about cancer is that
it
it
never
just
stops growing.
keeps yeah,
Yes.
it keeps growing. Yeah,
So,
because all
yeah,
those checks and balances of the cell have been taken away. And
exactly.
so if you're
Exactly.
dealing with something that's just a little bit dysfunctional but not cancerous yet, it's still a regular cell, right?
Still just a regular cell.
Just a regular cell. Oh my goodness.
Yes.
So people in the chat room were asking what the quote is on the poster behind you in your room. It's Ruth Bader Ginsburg.
It is. It is that. So I was awarded a career development award in honor of Ruth Bader Ginsburg, who did die from pancreas cancer. And so the quote was given to me as part of like winning this award, starting my lab to fight for the things that you care about, but to do it in a way that lead others to join you. And so that's kind of one of the lab mottos. Yes.
It's very inspirational. And it seems as though, you're not just, browbeating the people that you're working with into wanting to work with you on this. And then,
Yes. It
yeah.
is a shared passion. Yeah.
Yeah. And have you ever thought, I mean, do you feel like your background in music and your interest in the arts has given you a different perspective on your research directions and how you approach things?
Yeah, I think that a lot of people think that in order to become a scientist, you have to be like really book smart or like good at math or chemistry. I failed math. Everybody, I was not good at math. Struggled with chemistry and physics. Put me in a lab, I excelled because I have that
Mhm.
creativity and a very meticulous, like, receipt of data. And so I think that
Mhm.
for me, like I had a non-traditional path, but a lot of the creativity I used in like, I played the violin, so I did a little virtuoso work. That requires a lot of creativity, requires a lot of mental freedom and breaking out of
Yeah.
constraints of what you think is or is not possible or would
Mm.
not work. So sometimes like in science, we kind of box ourselves in. And it really requires a changing of perspective or a diversity of minds to kind of be the most innovative. And I've certainly found that's true. And that's something, a model that Salk celebrates because we have
Mhm.
a lot of science and music series. And so I
Yeah.
think it is something that is well recognized that without that creative spark, you know, a lot of our progress, a lot of our discoveries would probably be incremental.
It's the creative spark, but then also the motivation to continue, right? In the face
Motivation,
of
yes.
failure, which happens so often in science.
So we have this bug in the lab that says experiment, fail, repeat.
Yep.
So, yep.
Just get up the next day, keep
Just
going,
keep
and
chugging. Yeah. Yeah.
yeah.
Yeah.
So where do you, are y I mean, the disease in a dish organoid work, you can continue to focus strictly on the pancreas and that model system? Are you looking at trying to work with others or collaborate on other organ systems and transfer
Absolutely.
it?
I mean, so like, the organoid field is massive. And so
Yeah.
I would say that like, we're using a system that's really built by Toshiro Sato when he was in Hans Cleaver's lab. And it started off as studying the intestine, and then it
Mhm.
moved into like, the stomach, the pancreas, the liver. And so I'm primarily a GI organoid person, and we can grow organoids from all of those organs. And the reason why we're really interested in doing that is because we also want to be able to use normal organoids to find safe and effective doses of therapies. Because as much as we want to treat this, we absolutely do not want to impair normal organ function. If anything, we want to improve it.
Mhm.
And at least in my pancreatic cancer work, dose limiting toxicities are a major hurdle. It's something where even the latest breakthroughs in pancreatic cancer treatment are these RAS inhibitors. They come with
Mhm.
horrible side
Side
effects. And
effects.
so,
Yeah.
just having a more holistic view in our research of, it's actually quite easy to kill a cancer cell in a dish, but killing it in a person before you kill the
The
person
person.
is actually quite challenging. So,
Yeah. Thank you for working on all of this, and
oh,
thank you. Thank you so much for your time tonight. Is there anything else that you'd like to add that we maybe haven't covered, or that you was there
just
anything?
shout out to the lab and the team. Really, I just
Yeah.
want to say again, we couldn't do this without the patients that trusted us with their specimens, and also the clinicians, because it's no simple thing after having an 8-hour surgery to remove somebody's pancreas, then to like go back to the lab, put it on ice, FedEx it to us, and like send it on its way. It adds time to their days, and I know it's not something that is trivial. So big shout out to them for making this possible.
Yeah, for everyone. And thank you so much for your energy and the work you do to connect people over something that is essential. And
Appreciate it.
yeah, thank you for making a difference. We will have show notes and links to Dr. Engle's lab on our website. Thank you so much once again. It's been just wonderful speaking with you tonight, Danny, and I can't wait to hear about where this research goes in the future.
Yeah, I appreciate it. Thank you so much for having me. It's been fun.
Thank you. Have a great night.
Thanks, you too.
Good night. Everyone, that was Dr. Dannielle Engle from the Salk Institute. Her lab is the Engle Lab and focuses on the pancreas and pancreatic cancer detection and treatment, the early detection and treatment. And of course, we will have, like I said, links on twis.org. Everyone, we have a few more stories to come. I want to take this moment to say if you are enjoying the show, head over to twist.org, maybe click on the Zazzle link or the Patreon link. Patreon is how our show is supported. And we really can't do this show without people supporting us, just like you. Thank you so much for your support. And thank you for joining us for another episode of This Week in Science. I don't have music for Blair to come back to, so I'm just going to move on to my last couple of stories. I want to say once again, thanks to Danny Engle for joining me tonight. Who knew? I mean, a lot of people do know, but how many of us knew how important the pancreas is for so many things, and the work that it does for us every single day, and the work that Dr. Engle is doing to help personalize therapies is potentially going to be it's a sea change. I mean, I think this paper and the work are really a step forward in this direction. Yeah, people saying she was inspiring. Yeah, great info. And yes, many, many thanks to her team. So she said she works in the general GI tract. And so I'm going to move to my first of two stories to close out the show tonight, which are related. I'm moving away from the gut to the brain. A study out of USC Dornsife College of Letters and Science Research led by Scott Kanoski, published in Nature Communications this last week, was looking at, or actually this last month, was looking at the vagus nerve in rats and how it is linked to memory and the hippocampus. My favorite area of the brain is the hippocampus. And the researchers published their open access article in Nature Communications, finding that the vagus nerve, people talk about our vagal reflex, like the vagus nerve is responsible for calming us down. The vagus is very important in excitement or digestion. And the vagus nerve relays metabolic information between the GI tract and the brain. And people are like, okay, memory, whatever, let's go in there and it's working. But this study found a direct connection in rats between vagus nerve mediated acetylcholine release in the dorsal hippocampus. That's the back of the hippocampus. Well, it would be like back of the hippocampus. The dorsal hippocampus from what are called medial septum neurons. So basically the vagus nerve coming from the GI tract is coming up and stimulating your hippocampus. Hippocampus related to memory. And so they started looking at what the rats were eating and what was being remembered. And they discovered that, oh, hey, this is exciting. The stomach, your stomach, when it gets junk food or sweet treats, and I'm extrapolating as they did a little bit, but in these rats, when they were given sugar, when they got sugar or fat, the hippocampus got super excited based on that vagal nerve stimulation, and there were really strong memories that were produced. And so sweet tasting but low calorie treats did not have the same effect. And so the stomach is talking to the brain going, you gave me sugar or fat, and this was awesome. Remember it. And the researchers are saying, hey, this is an important potential pathway evolutionarily, because if you find a really nutrient dense, valuable food, you want to remember where it came from because maybe you can go back to it again. They did find, however, that when the rats consumed this type of diet early in life or for long periods of time, that the relationship weakened. And so the memory formation from these rich, sweet, fat treats, they become less strong, right? So it's the same kind of thing. If you're always having a chocolate bar every single day, that chocolate bar is not going to be as exciting a thing. Whereas if it's the one special Santa chocolate that you got on Christmas, or the lollipop that the Easter bunny gave you, or whatever it happened to be that you remember from your childhood, those were special experiences because it was a one-off kind of deal. It's not all the time. And so it's really interesting that diet and consistent diet versus these treats can have a disruption to the acetylcholine signaling in the hippocampus. And so they're also suggesting that for humans, constant exposure to junk food might disrupt or disturb the connection between our gut and our brain, so that we lose that special relationship and food stops having as much meaning after a certain period of time. They've also shown that disruption of acetylcholine signaling in the hippocampus, according to Kanasaki, the lead researcher, is one of the earliest neurochemical changes in Alzheimer's disease. And by revealing that this system is boosted by gut signaling from the vagus nerve, there are potential novel therapeutic targets that could potentially be leveraged to explore vagus nerve-based approaches. So maybe vagal nerve stimulation could help to ameliorate some of the problems related to Alzheimer's disease, which is really interesting. Diet has so much effect on us, and now we're finding more evidence of these direct connections between our gut and our brain, and it's very exciting. I think it's very exciting anyway. But yeah, they were able to find out that when rats ate too much sugar food, the brain, the vagus nerve stopped signaling as much and it didn't make as big of a difference. So, of course, we're going to extrapolate to humans. Moving on to my last study, which is now just the brain and has nothing to do with your stomach, unless, of course, it's maybe learning how to do sit ups or learning how to, I don't know. I'm not going to try and make something up on the fly here. This is the brand new study out of PLOS Biology, again, open access. So if you're interested in reading it, you'll be able to get access. Researchers published their work looking at the human brain. And I want to say, I think this is the first time I have ever seen this approach to trying to see changes in the brain as a result of learning. Historically, we have to slice things apart. The reason we use mice and rats and other animals is that being able to study their brains comes with a little bit less of an ethical issue than using human brains. So these researchers published their study, Learning Engages Transient and Sustained Cellular Mechanisms in the Human Brain. In this study, they used a new technique based on advanced fMRI diffusion tensor imaging. So diffusion tensor imaging is one of the newer forms. It's been in use for quite a while, though, of these MRI techniques of imaging active areas within the brain. And the diffusion tensor imaging is able to actually see changes down axons and dendrites within the brain. So it's been able to help researchers start actually seeing the changes in like movement of stuff along pathways from the cell body of a neuron to another part, a connection at some place. And so the diffusion tensor imaging has really given researchers a new view on activity within the brain at a very cellular level, as opposed to just, oh, this part of the brain is excited versus the other. Okay. And so the researchers, they've used an entirely new form. Well, it's not entirely new, but what they have done is they've combined ultra high gradient, so very high resolution, diffusion MRI with what they call SANDI. And SANDI is soma and neurite density imaging. And so they use magnetic resonance imaging and they use this mathematical model to distinctly divide different categories: cell bodies, the cellular extensions, the neurites, and then the extracellular fluid that's around all of the cells. And so based on a lot of math and this really high resolution diffusion magnetic resonance, they're able to go, oh, the cell bodies are active. Oh, the dendrites are active. Oh, wait, the extracellular fluid is changing or whatever. They're able to get really specialized view on the brain. Okay. And so this is where it gets extra special exciting, because the researchers were able to have humans, adult people, learn just a simple task: motor sequence learning, tapping on little numbered keys. 1, 2, 3, 4, 5, 4, 2, 4, 1, 3, 2, 4. And you learn it once and repeat, learn it till it's memorized, rest, do it over again, and see how the brain changed. And so they used this technique of doing this SANDI diffusion magnetic resonance imaging twice over a 24-hour period and testing again. They were able to show where specific changes were happening within the brain. And I'm going to say, normal brain imaging, we're like, okay, great. Look at that. It's prefrontal cortex. Or, oh, hey, it is the motor cortex of the brain. Normal, 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 normal. That's fine. But what they saw actually were changes in the cell bodies. They were able to differentiate between actual changes within the cells and the dendrites, the neurites, the axons of these cells. And this is where this is just the exciting part, because you don't get this kind of detail, and they were able to show that there were transient and persistent changes in the microstructure. So the hippocampus, say, that's one part of the brain that's responsible for memory, right? Okay, you're learning something. What changes are happening in the hippocampus? They showed that when you're learning something, after you've started to learn something, the cell bodies in your hippocampus swell. They get bigger. So you're actually increasing the size of the cells, neurons in the hippocampus of your brain when you're learning stuff. And they showed that in the primary motor cortex, swelling in the cells, there over 24 hours in the primary motor cortex, there was an increase in the size of the neurites, the dendrites, the axons. There was plasticity happening within a 30-minute to 24-hour period. And then persistent changes, they found there were real changes that lasted in the precuneus and the posterior parietal cortex. And that, again, after 30 minutes, there were changes in the cell bodies. There were changes in the dendrites, the axons. These are the changes that lead to you learning. There are actual physical changes in your brain, that lump of pink jello goo that's inside your skull, it changes. Little tiny cells are growing and shrinking and growing and shrinking when you're learning stuff. And that leads to permanent changes over time, which to me, this whole study made me so excited because I have not seen anything like this before. This microstructure work is totally new. Someone else out there who's working in neuroscience more consistently might say something different, but I think this is a huge step forward in our ability to measure certain aspects of change in response to stimuli within the brain in living human beings without having to slice into people. And this is a huge, huge advancement for the field of brain science. It's so exciting. This to me is like one of the most exciting things. We don't have to slice something up. Oh my gosh. It's very exciting. So, I don't know if I can say exciting enough to get it across to you. I think it is. But anyway, that is my story, my last story. I didn't bring a ton of stories tonight because I'm on my own. And also I had that wonderful interview with Dr. Engle. So if you have questions about what happens in the vagus nerve staying in, I see your post there, Paul Disney. And yeah, Pepsi Colas. I think it is a very neat find. It really gives me some optimism about the technology advancements and the potential for their use in understanding our brain and how very short time period changes might lead to the longer time period changes, and being able to see how physical changes in the brain might correspond to behavioral changes it's something that our brain, our head, it's a black box. And so anyway, I'm going to end the show and tell you to sleep on it because you learned something. And so go to sleep with all your swelling brain cells and enjoy your dreams, and know that you have changed your brain for the better by coming and being a part of TWIS tonight. Thank you for joining me tonight. I do appreciate it. Hopefully next week Blair will be back. I don't see any reason why she won't be. But we are going to be back again next week. Lon, we already started the show. I'm sorry. Yes. Thank you for being here. Everyone in the chat rooms, thank you for being here and being a part of the conversation as we were going through the show. And I have a few more thank yous to make, as I always do when it is the end of the show. Thank you to everyone who helps with the show. Thank you, Fada, for all of your help with show notes and show descriptions and the work for YouTube and everything that you do. Thank you, Gord Arnlore, everyone who helps keep our chat rooms amazing places. I love seeing the great conversations that happen in the chat room and all the things you have to say. Questions, jokes, all welcome. Identity 4, thank you for recording the show. And thank you, Rachel, for your work editing the show. Thank you, of course, to our Patreon sponsors. Thank you to dun, 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 Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Artem, Arthur Kepler, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Derek Schmidt, Don Mundus, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis 104, G. Burton Lattimore, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Rattaswamy, Johnny Gridley, Ken Hayes, Kent Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow, Mark Jenkins, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Richard Badge, Robert Norland, Robert W. Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence Lamb, Shoe Bruce, Stephanie Embry, Stephen Alberon, Steve Leisman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, and Vegard Schefstad. Thank you for all your support of TWIS on Patreon. And if you would like to support TWIS on Patreon, head over to twis.org and click on that Patreon link. Anyone supporting us at $10 a month or more will have their names read and hopefully not mangled by me at the end of the show. We'll be updating our monthly Patreon list for reading next week's show, because we'll be in August. It's time for updating the list and who's going to be the new person on the list. I can't wait to find out who's it going to be. On next week's show, we're going to be back Wednesday, 8 p.m. Pacific time-ish on our Twitch, YouTube, and Facebook channels, broadcasting live where you can watch and join the chat or find us later just if you're subscribed. If you want to listen to us a podcast, of course, subscribe to us wherever you find your podcasts. Look for This Week in Science and subscribe. Get your friends to subscribe too. This is the kind of help we need. Tell people they are missing out if they are not joining the show on a weekly basis. For more information on anything you've heard here today, show notes and links to stories are available on our website, twis.org, where you can also sign up for the newsletter that will arrive someday. We love your feedback. If there's a topic you want us to cover or address, or a suggestion for an interview, please let us know in one of our social media accounts or send us an email. Oh, I just remembered I got an email from somebody and I forgot about it. I'll bring it to next week's show. Just put TWIS in the subject line so your email doesn't get spam filtered into a faulty CFTR channel, because then it won't help to make anything easy. We look forward to discussing science with you again next week. And if you learned anything on the show, please remember, it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. 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 This week in science.